Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show. Today bringing you another fascinating guest who is helping to create a better tomorrow for many people around the world. Today we have the honor of being joined by Dr. Sanjoy Dutta, who is Chief Scientific Officer at the Juvenile Diabetes Research Foundation, or JDRF International, a nonprofit organization. It funds type 1 diabetes research, providing a broad array of community and activist services to the type 1 diabetes population, as well as actively advocating for regulation favorable to medical research and approval of both new and improved treatment modalities. Dr. Dutta oversees all of JDRF's efforts to cure type 1 diabetes and improve the lives of those living with it, which includes various applications of technology, including beta cell therapies, immunotherapies, glucose control, and related disease complications. He is also responsible for international partnerships with various world-leading government, non-government foundation, and commercial organizations. He joined the JDRF back in 2009. Prior to that, he was the Associate Director of Translational Medicine and Clinical Biomarkers at Bristol-Myers Squibb. He also served as a Principal Scientist of Cardiovascular and Metabolic Diseases at Hoffmann-La, Roche. Dr. Dutta obtained his doctorate from the Department of Biochemistry and Molecular Biology at University of Southern California. He was a JDRF Postdoctoral Fellow in the Department of Cell Biology and the Joslin Diabetes Center at Harvard Medical School. A lot of interesting things to talk about today. Dr. Sanjoy Dutta, thank you for taking the time to come on the show.
Thank you, Ira, for the invitation. I look forward to this.
Yeah, us too. And, I'm really interested. I typically hand the floor to our guests, which I'm going to do with you as well, to sort of talk just a little bit more about your background and sort of how you just got into your interests in biochem and molecular biology and so forth. But I also noticed sort of very early on in your career, neuroscience. How did you ultimately make the transition from neuro into the metabolic space? I think that'd be interesting to start out.
Yeah, I mean, I did my education mostly in India at the time here, and I completed my master's there. Then I moved over here. So, I had a background in chemistry as an undergrad, and then as I went into my master's program, it was a biochemistry major, but then we had to specialize in one of the other additional areas, and I had chosen at the time of neurobiochemistry. I wanted to become a neuronal scientist, but the career took me in a different direction, which is not vastly different, especially in diabetes where there's a significant amount of neuropathy, but I'm not a neuronal scientist today.
Sure.
Yeah. And then, I've as you've read my bio, I've been in this field of medical research, basic research, and some translational research for the last 20 years or so, both through my training as well as in my careers in the pharmaceutical industry, as well as now at the JDRF. I've focused on both type 2 diabetes and type 1 diabetes and metabolic disorders that come with it, such as, obesity and comorbidities or complications of diabetes. So, and at JDRF, obviously, we've been focusing almost entirely on type 1 diabetes. And we'll talk more about the promises, the promise of today and the hope of tomorrow.
Excellent. Yeah, I mean, JDRF, obviously, it holds a place in a lot of our minds, especially here in the U.S. I've done the walks here locally in Philadelphia, and it's one of the medical foundations that's been around for quite a while, for a few decades now. And obviously, it's morphed as time has gone by from sort of the activism, but obviously you're actively sponsoring, promoting research on many fronts, and you have venture capital activities now. You have investment in basic research. Talk a little bit about how the organization sort of started out, of course, but also how it's evolved over the last few decades, if you would.
Yeah, Ira, we have been around for over 50 years, and so you can imagine significant progress and evolution of the organization, along with keeping pace with the evolution of the science that has happened in the world of type 1 diabetes here. And I don't think I can cover it in a few minutes, but I'll do the best I can. But
take your time.
One thing I pride myself in every day I work at JDRF here is that we were founded by families, that had type 1 diabetes directly in the family. And we were founded, as you said, as an organization to raise awareness and education in type 1 diabetes research, which 50 plus years ago was minimal, if any, at the time. So I pride myself in that. Even to this date, we are governed by our board, our various committees that are directly people that have a type 1 diabetes connection. Either they themselves have it or their immediate family has it. So it is the passion of type 1 diabetes is kind of woven in the DNA, in the infrastructure of JDRF, and I pride myself in that. In terms of evolution, when JDRF started, it was actually called JDF, Juvenile Diabetes Foundation. And then eventually, within a few years, it was realized, many things were realized. I'll highlight just a couple of them. Sure. The biggest thing that was realized is there was a huge gap in research. There was not dedicated research by any organization in type 1 diabetes, let alone the knowledge and the awareness, whether it's federal resources, whether it's other private resources or other foundation resources such as ours. It was practically nothing, several years ago. And so our founding members decided to put the R in our name to emphasize the need for research to understand the disease, to understand ways of intervention that we can either prevent, cure, or treat type 1 diabetes and its myriads of complications here. Our mission is to develop life-changing breakthroughs that can accelerate preventive, curative, and better treatments for type 1 and its complications. Our vision actually is a world without type 1 diabetes, where we can eradicate it from the next generation. The second stream of evolution at the time, in the beginning, was soon realizing that we can't be US-centric. We started in the United States, but we soon realized that this disease is, it's beyond borders. It crosses borders. So people around the world are affected by it, and we have to support the best research anywhere in the world. We have to be an international organization, and soon we set up our capabilities to support research anywhere in the world, as long as it merited the quality of peer review that we usually use as our litmus test. And we had established a physical presence with our affiliates in several countries outside the United States, but our research funding is not restricted to only where we have physical presence. For example, in Canada, the UK, Israel, Australia, and Netherlands. In the past, we've had other affiliate presence, but we currently are funding in over 20 countries, and historically we have funded in over 30 countries. So we can fund research anywhere we can. So initially, those were the two main evolutions or advancements or enhancements to JDF, to JDRF that happened several decades ago. But since then, we have obviously, as we supported more research, as we learned more about the basic biology of the disease, how the disease progresses, who is at risk, and so much we have learned, and I can talk about it, Ira, in a few minutes, but there we have realized that basic research is the foundation of understanding the disease. It's very critical to know the genetics, to know what are the non-genetic environmental triggers of the disease, to know who gets it, when one gets it, why is it severe in one person versus another. Sometimes two siblings, the disease may present very differently in two siblings within the same family. As we supported those, we realized that in parallel, we need to really set a translational tone to what we are doing, because our mission ultimately is people with diabetes doing better. If people with diabetes are not doing better, we haven't done our job. we haven't done our job. And so if that's the only metric we judge ourselves with, and for that, we needed therapies developed. So we entered into partnerships with several organizations, including the commercial sectors, because they are the ones that develop the devices, the drugs, the biologics, the cell-based therapies, so on and so forth, and combinations of those. So we need to impress upon them that this is a disease that can be managed, that can be treated, that can be cured, and hopefully that can be prevented also. And we needed to work with them to not only show them that there's a way, but also de-risk their investment in this. So we have a lot of industry partnerships in addition to our significant and major funding in the academic sector. We were fortunate. We were fortunate that in this journey, we could bring along partners from federal organizations, from other not-for-profits, private and public charities. And these federal organizations were not just restricted to the United States. They are in Europe, UK, Australia, Canada, and other parts of the world there. And then more recently, adding one more tool to our toolkit, to our, in how we work and where we work, there was a big gap in venture capital investment. Yep. And that gap was realized several years ago, and there was practically zero investment in the VC world in type 1 diabetes. And that's when the JDRF T1D fund was born about 4 or 5 years ago. And that has also had significant success in bringing in not only resources to support translational research, but also a big think tank to go with it. So we deploy these various tools of funding research in parallel developing advocacy efforts, so we cover the entire pipeline. So there's been a lot of evolution. As I told you, it's a big topic, Ira, but I kind of gave you the broad strokes of where we were, then where we were about 30 years ago, and where we are today. And we realize that we are a leader in type 1 diabetes in the world, but we are fully cognizant that we cannot do it alone. We will need all of these partners to work with us to get us to our goal.
Yeah, it's really fascinating. Obviously, thinking of how we. You've published a lot on this too. How we, in the past, obviously have looked at type 1 diabetes sort of with this one lens. Okay, here are these beta cells. They are no longer producing insulin. They're being destroyed. Fine. But now, as you were mentioning, you have sort of the whole continuum of the comorbidities that follow. And then you've published a lot about what's happening upstream. And one area that sort of is near and dear to you is sort of the immune system. And you're very, you've written about, look, the longer that we could keep the functioning beta cells functioning and keep away that autoimmune destruction, the better. I mean, that's a tremendous benefit. And you've also written about disease stratification, that there's a lot of other things to look at when we're looking at these patients than the HbA1c, in terms of the pre-symptomatic component of how this disease presents. Your organization was instrumental in funding this TrialNet study with teplizumab, a humanized anti-CD3 monoclonal antibody, once again looking at how to sort of keep the immune system at bay in T1D. Talk a little bit about sort of this whole area of managing the autoimmune component of type 1 diabetes and how the tools, as you were mentioning in some of your papers, to understand the progression of the early disease have become so important.
Yeah, Ira, that's probably one of the most important questions that we will be discussing about is in understanding the disease. We go in any disease, especially in chronic diseases, and I've worked in a couple others in the past, understanding the genetics, the basic biology and the basic pathology of it is critically important. One of the seminal contributions in the field, led by JDRF, but many, many others, has been staging the disease. Right now, type 1 diabetes is diagnosed when one reaches a certain value of a hemoglobin A1c, which is a circulating marker of a glycosylated protein, hemoglobin, that reaches a certain threshold, and what percent of that is glycosylated is measured as whether you have diabetes or not, both type 1 and type 2. That is somewhat of an artificial number, but that's the number one goes with, and that's when one is put on insulin and one becomes insulin dependent for the rest of their life. The seminal contribution here is, and JDRF has the firm conviction, and many others in the T1D community, is you actually have type 1 diabetes before you've been told you have type 1 diabetes and been put on insulin. Yep. This staging has been very critical, not just to understand the disease, but to develop preventive strategies. And what we have done in a nutshell is with research efforts supported by JDRF and many others, including the NIH, is that stage the disease into what we call stage 0, 1, 2, and 3, where stage 3 is when you're diagnosed with type 1 diabetes and you're put on insulin when you reach a hemoglobin A1C level. And there are stages 4 and what we sometimes call stage 5. Stage 4 is a person living with type 1 diabetes for years and decades and completely insulin dependent, lost most of the functional beta cells, if not all of it. And stage 5, it's not well defined, but is when you have type 1 diabetes and also develop some complications. Let me go back to stage 1 and 2. This is before you're diagnosed with canonical type 1 diabetes.
Okay.
In stage 1 is where you start showing signs of autoimmunity in your blood. You develop antibodies, and these are measurable. And so we can measure several antibodies in the blood that show signs of autoimmunity already kicking in. So you actually have the disease before you know you have the disease.
Mm-hmm.
You still have good glucose control. You're not what we call dysglycemic at this stage, in
Okay.
stage 1. Then as the disease progresses, as your autoimmune system, your immune system, which is autoreactive, starts destroying the only cells that produce insulin, i.e. the beta cells in the pancreas, then you start showing signs of glucose, loss of glucose control,
Mm-hmm.
sometimes called dysglycemia, sometimes called glucose intolerance. You still haven't reached the threshold hemoglobin A1C, which is diagnostic of type 1 diabetes, but you're already showing imbalances. You're showing a lot of glucose variability through the day. You're showing when you're challenged with a meal, you're showing very high levels of glucose curves there. That's stage 2, where you have the autoimmunity plus a level of glucose imbalance there. And then when you reach a certain nadir of your beta cells, where you no longer can cope with the remaining beta cells in your body, and that varies a lot from people to people. In some people, it may be 70% of beta cells lost, in some maybe 50, in some maybe 90. That's when you need exogenous insulin injections, or through a pump to supplement the loss of insulin production within your body, within your beta cells. And that's stage 3. So stage 1 is where your autoimmunity has kicked in, but your glucose levels are pretty good. Then stage 2 is where autoimmunity continues to destroy beta cells and glucose levels start showing imbalances and signs of a storm to come. And then stage 3 is when you absolutely need external insulin to control your blood glucose levels. This autoimmunity process, and we right now know of many genetic factors that can contribute to it, but what we've also learned, Ira, and this is very critically important, is that 90% of the people, 90, not 19, 90% of the people newly diagnosed with type 1 diabetes do not have a family connection.
Okay.
If you have a family connection, which is approximately 10% of the people, you are definitely at a higher risk, 15-fold high risk. And so there is a huge genetic component, but that's not the only factor. There are environmental triggers, which we are still trying to understand, that can trigger type 1 diabetes in an individual. 90% of them get it through this. Another critical consideration to point to make here, Ira, which 30 years ago, 20 years ago, I would not have believed, is that it's no longer a juvenile onset disease only. 50%, 50% of the people newly diagnosed with type 1 diabetes are actually above the age of 18. So a lot has changed. Just as JDRF has evolved and pivoted and adjusted our strategy, the knowledge around the type 1 diabetes disease has also changed significantly. And therefore, we have had to adapt that. And how can we intervene to curb the immune system? It's not a matter of shutting down the immune system, because that would be derogatory.
Sure.
How can you curb the immune system? What are the factors within the immune system? It's a very complex system, like the neuronal system. The immune system and the neuronal systems are probably the most complex in our body. There are so many networks that are interconnected, and you can't really put your finger on the pulse for one particular thing, one particular pathway. Okay. All that said and done, how can we curb the immune system so that we reset the balance while we develop strategies to produce the beta cells in the body? How can we regrow, regenerate the beta cells that are lost in the body? So we believe a cure will likely be a combination of an agent that resets the immune system along with an agent that replenishes the lost beta cells in the body. We believe that would be a cure. And just to finish up on that part of our research efforts, another important way to provide curative options or insulin independence to the people is to grow the beta cells outside the body in a petri dish, in a flask. For example, stem cell based or through other sources of beta cells. If we can grow them, which we know how to do right now, and we have 4 or 5 clinical trials ongoing at the moment, and you've heard about Vertex and Biocide and Cernova and other companies out there. So then we can transplant these beta cells from outside the body into the body and provide insulin independence and a whole milieu of metabolic benefits to the people. So there are that's why we refer to cure in plural, cures, because there will be multiple options, multiple ways of curing the disease. And given the disease is very heterogeneous, definitely it's not going to be one size fits all.
Sure.
And we have to have these options on the table here. So that's more on, to answer your question about autoimmunity and staging the disease and disease modifying. And just to finish up, teplizumab, the drug you named, has long been supported by JDRF. I would say about 30 years of research by JDRF has funded. Many other funders have funded. TrialNet is a consortium funded by the NIH, National Institute of Health, through what we call a special diabetes program funding. This is federal appropriations from that goes to NIH that JDRF is the primary advocate for. It's a significant support that goes to the NIH to support efforts such as TrialNet and many, many other critical research programs that have developed important therapies for people, one of which is teplizumab. And that's currently under consideration for delaying the onset of disease by 3 years, which is significant for people that are at risk of developing the disease.
Sanjoy, you mentioned, obviously this potential holy grail situation. You have the control of the immune response and then the cell replacements. And I had the opportunity to talk about some of these different pathways that are out there. I've talked to people like Camilla Riccordi at University of Miami with the beta cell transplants. I spent some time with Dr. George Giddy at the University of Pittsburgh. He's working more on the trans differentiation side of things, how we can get these other cells, the alpha and the delta and so forth, that are there, and via genetic technologies maybe make beta cells. And then I recently had the CEO of Novo Nordisk on. They just put $300 million into a couple different centers looking at various different applications of stem cells in terms of IPS and other tools. What are you personally hot on when it comes to the stem cell area? Because stem cells means a lot of different things nowadays. What stem cell technologies are you, have unlimited funding? Are you putting, would you going to put most of the resources into, you think?
All of the above. All of them. Yeah. There's promise in all of the above. Whether you take an embryonic or an adult stem cell source and you convert that into a beta cell or a beta cell-like cell that produces insulin, and then you transplant it, so that's there. There's also what we call inducible pluripotent stem cells, which is taken from an adult, like a skin fibroblast or something else, and you can convert that into a beta cell. And all of these have significant promises. We're also supporting research in what we call xeno source or non-human sources, such as swine sources and others, and because it's very close to human biology, in trying to produce these beta cell sources, and all of them have significant promise. One thing you didn't bring up, but I'll mention why we have so much faith and we need to try all these options, is because we have a cure for type 1 diabetes today. We have a cure, and that is islet transplantation. That's islet. So people that have received an islet transplantation, and this is actually an approved protocol in many countries, unfortunately not yet in the USA, but there are islet transplantations done along with kidney or renal transplantation in the United States as well. There are people 5, 10, 15 years out that have received islet transplantation, 1, 2, or 3 rounds of transplantation, that are not only doing extremely well with respect to their diabetes control, but are also getting many other glucometabolic benefits beyond that. The caveat, as you can imagine, Ira, is 2. the good news is there's proof of concept that you can cure type 1 diabetes. You can make the people insulin independent. You can get rid of the severe hypoglycemia that was debilitating their lives, both clinically as well as from a lifestyle perspective. There are two huge challenges. One is a simple supply-demand imbalance. The supply of cadaveric islets is obviously very, very, very limited, and the yield is extremely poor. And the second, and the demand is obviously more. There are, 1.5+ million Americans just here, and then outside around the world, many more million people with type 1 diabetes. So there will never be a supply-demand equilibrium if you only rely on cadaveric islets. Point number one. Point number two is that, like in any transplantation, liver, heart, kidney, lung, and pancreas or islets, you'll be on chronic immunosuppression throughout the rest of your life. And that is not a fun situation. And so therefore, trying to develop these novel sources of insulin-producing cells, wherever they come from, and then protecting them inside the body once implanted, and so that one doesn't have to have chronic whole body immunosuppression the rest of the life, is a main goal of JDRFs. And we are feverishly pursuing every option that's on the table. And all of these options have promises. And we can't pit one against the other, because the evidence for one is not necessarily, we are yet not able to decide as to one pathway of stem cells versus iPSCs, which is also stem cell inducible stem cells, or it's a porcine source. We don't have enough information today to make a decision, and that's okay, because we can try we can advance all of these options.
It's interesting because you brought up Xeno, and I don't know if you saw the episode. I just had David Cooper on the show about 2 weeks ago from Harvard, and I got to say, thinking back, because he wrote a book about 20 years ago on xenotransplantation. I never thought we'd get this far, and now the things they're doing with pig hearts, the amount of genes they can modify to get sort of the autoimmune, the histocompatibility stuff out of there, deal with the retroviruses. I mean, it's really fascinating. Keeping that in mind, when it comes to are xenotransplants of pancreases something that is sort of, I know obviously they're focusing on hearts nowadays, and I haven't done that much of a deep dive into the world of xeno and pancreatic islet transplants, but is that a hot area in its own right? And then taking that, coming from to the pancreas, here we have a natural but porcine-derived pancreas. Talk also a little bit, if you would, take us into the world of the artificial pancreas and some of the things you're looking at there. So two-part question, kind of disparate, but...
Yeah, that's a huge success story. So finishing up on the xenotransplantation, yes, there is a lot of promise. There are some safety hurdles that we have to cross, not insurmountable, in terms of the xeno-specific viruses that could be deleterious to the humans. And so they are not insurmountable, as you mentioned already. This gentleman, 68-year-old gentleman, has been rescued with a heart transplant from pig, and they're already pursuing a kidney transplantation using xeno sources there. So it's not unfathomable, it's surmountable, but there are some barriers we have to overcome. Artificial pancreases, or sometimes called automated insulin delivery systems, have probably been the biggest success. So JDRF started this program about 15 or so years ago, and at the time it was space technology, space science, to be able to do it. And the interesting piece is that we've not only been successful, we have huge success here. So for viewers that are going to listen to this or see this one here, just to level set everyone here, artificial pancreas is described right now as a component system which has three parts to it. One is a pump that delivers insulin to the body. It's usually subcutaneous pump. Second part of it is a continuous measuring device that measures glucose called CGM. And the third part is what we call the brain of the artificial pancreas. That is the algorithm that cross-talks between the pump and the CGM. An artificial pancreas system has to have all of the three components to it: the pump, the CGM, and the algorithm. That's what constitutes an artificial pancreas. So we right now have 3, and if you count separate pediatric approvals, 4 or 5 approved artificial pancreas systems available in the United States, and a fewer than those numbers around various parts of the world. What have they done? They have provided extremely good outcome in people with diabetes, an outcome as measured by not only hemoglobin A1C, which we already discussed, but also other very critical factors for people with type 1, such as reducing the variability of glucose, the swings, the upswings and the lower swings that one experiences throughout the day. It also provides good time in a desired glucose range. It has reduced hypoglycemia, a dangerous outcome of type 1 diabetes. It has reduced that significantly. So artificial pancreas technologies have provided significantly improved clinical outcomes in people with diabetes in terms of good glucose control. Another important thing to remember, this is equally important, and I can't rank order the two of them because they're both number one. It has reduced the burden of this disease. Let's not forget, this is a 24/7, 365 disease.
Sure.
A person with type 1 diabetes on an average makes 300 plus decisions in a day. A lot of these have been alleviated with it has not been completely removed. A lot of these have been alleviated with artificial pancreas technology. Let's not forget, it's what we call hybrid closed loop. It's not completely closed loop yet. It's not completely automated yet. There still needs to be human intervention, some calculation done for meals, for example, when you need an extra bolus of insulin. You need some, when you exercise, especially severe exercise, there needs to be some human adjustments still to it. And JDRF's goal is to try and completely close the loop, completely make it automated so that the human component is removed, the human burden is removed, and the human error is removed. And so the next generation of AP systems will hopefully get us there, but there are some things we need to develop before them, such as ultra rapid acting insulin, so on and so forth. So, so today's artificial pancreas systems are really, really good. They are not fully automated. They are semi-automated systems that still require some human intervention.
Got it. Got it. Sanjoy, at the beginning, I mentioned some of the other activities that have come online at JDRF in recent years, one being your T1D venture capital fund. You recently also funded this $14 million JDRF Center of Excellence, University of Michigan. Talk a little bit, if you would, about, obviously, if I'm a promising young entrepreneur, promising entrepreneur that has innovative technologies, what is the JDRF T1D fund all about? What are you looking for specifically there, and sort of types of investments you make out of the fund? And then, if you could talk a little bit about University of Michigan, what sort of goes into creating a new center of excellence? Because that's not a small figure, $14 million. How do you come up with some of these projects at the same time?
Yeah, absolutely. Very happy to talk about it. As I mentioned at the beginning of this call here, the JDRF T1D fund is a venture capital fund that takes it makes an investment in equity, takes an equity position, and then all the profits from those are circled back into research here. So it's not a classical VC fund as one would. And what they're investing in is translational science here, and how we can get these therapies across here. A lot of the research they fund, not all of it, often comes from what the foundation, what JDRF, the mothership, has funded in terms of our R&D process. And when you reach a certain stage of translation, there are some things that very naturally and organically are taken into clinical development or are transferred into a pharmaceutical or a biotech company. There are some that languish, that require some more research, some more R&D, and that's where the fund comes in here. That often, sometimes, is referred to as a valley of death. But regardless of that, that's where the fund can come in. They often galvanize a lot of intellectual resources, along with financial, of course, bringing in researchers there, and then supporting a biotech company to move the research forward to the next point of inflection. And that could be making it ready for clinical trials, making it ready for the next stage of a clinical trial if it's already in the clinic. Some recent examples of the success of the JDRF T1D fund, and it's not just limited to these. One would be teplizumab that we discussed already, where they supported Provention Bio in the past. The second one would be supporting SEMA, which was a technology developed out of Professor Doug Melton's lab at Harvard University that JDRF, the foundation, supported the stem cell work. And then the small biotech company SEMA was formed in the Massachusetts area that the JDRF T1D fund then invested in.
Nice.
And then when the technology reached a certain point of maturity inflection, it was acquired by Vertex. So the T1D fund is no longer investing in it, but they carried this risk, this period of risk through support of SEMA. And now Vertex is obviously being very successful there in a clinical trial, and you've seen a lot of the news over the last 3 or 4 months. Regarding centers of excellence, JDRF has always been a big proponent of these centers of excellence. Now, what does that mean? We have multiple funding mechanisms. We often do grants as small as 1-year grants for $110,000 called innovative grants, where we just take an idea in and we provide some support research funding to prove if the idea works or not. We have multiple tools in our armamentarium. One is supporting very early stage innovative research, supporting trainees so that they become the future T1D researchers and clinicians that can carry the people pipeline, supporting proper academic grants, industry partnerships, dedicated partnership on projects for therapy development. And then this tool is Center of Excellence. What does it mean here? Where the scientific problem we are trying to address cannot be often solved by one individual investigator. It requires multidisciplinary, multiple points of attack at the scientific problem. For example, it could require cell biology. It could require expertise in metabolism. It could require expertise in systems biology. And oftentimes, a single lab doesn't have that diverse expertise. You need to bring in these people. You mentioned Center of Excellence at the University of Michigan, and we have currently about 5 centers of excellence that we are supporting. One in the Northern California area between UCSF, the Gladstone Institute, and Stanford University. A second one in New England, which is supported the Harvard Stem Cell Institute, University of Massachusetts, and the Joslin Diabetes Center. Then there's one in Canada and British Columbia. There's one in Perth in Western Australia. And then the University of Michigan, which is in one university. And there are different investigators tackling one core scientific problem, which is the metabolic dysfunction that happens in type 1 diabetes. Oftentimes, we get very laser focused and myopic on glucose levels. It's important because that's what goes awry.
Sure.
But in that looking for that tree, we miss the forest. There are many other metabolic pathways that are dysregulated or aberrant in people with type 1 diabetes. So having a healthy metabolic milieu will not only help the individual have a good glucometabolic control, avoid complications, but also have psychosocial benefits, also provide a very healthy environment when you do, for example, cell transplantation. When you're trying to implant stem cells into a person's body, if the metabolic milieu is sick, so to speak, the cells aren't going to survive. So the University of Michigan Center of Excellence focuses on this overall metabolic dysregulation in people with type 1 diabetes, and how that can be a central tenet that then can be that if you can reset that metabolic balance, how you can benefit other therapies, artificial pancreas technology, reduction in complications, potentially psychosocial behavioral benefits, and cell replacement therapies that can now have a healthy home to reside in when you do the implantation. So centers of excellence are intended for that. There are other critical components in centers of excellence beyond the science. For example, training the next generation of minds. Because we need to, as we support the therapy pipeline, we need to support the people pipeline. Who will be the next generation type 1 diabetes researchers? Who will be the next generation clinicians that will take over? So these centers of excellence have a very dedicated effort in grooming next generation scientists as they learn from more senior investigators and mentors and become our future leaders of T1D research and care.
Outstanding. Sandra, we've talked now about beta cell therapies, immunotherapy, glucose control, and complications. I want to loop back to something that you mentioned early on. That was the word triggers, and I have to. Obviously, it's 2022. There's still, as you were saying early on, there's still a lot of things about the early stage, the triggers of this disease we don't know much about, the unique genetics and so forth. But I have to ask, because you were quoted, and a lot of people have been asking you about this CDC study from a couple months ago. 2.5 million patients younger than 18 studied. CDC says 2.5 times more likely to be diagnosed with diabetes a month or longer after infection. At the time, your response to that was, we got to study this a little more. This does not look very good. But talk a little bit about what you're thinking about, what you found with this particular study, and then just if you could say a few things about potentially the viral component here in general, whether it's COVID or other types of infectious insults that may be very far upstream, but maybe somehow connected to some of the autoimmune stuff that goes on here.
Yeah, it's a great question. And the short answer is we don't know. So
cool.
Environmental triggers, while we say environmental triggers, and that goes back to my original comment about 90% of the people don't have a family connection, and 10% that have the family connection are certainly at a heightened risk. And also 50% of the people are getting the type 1 diabetes diagnosis after the age of 18, some in their 30s, 40s, 50s. And so all of these things, as well as a significant amount of research, has shown, and this also brings in the heterogeneity of type 1 diabetes. So specific viral infections have often shown to be triggers, particularly in Finland and some other parts there. Coxsackie B virus, which also happens to cause the ear infection, and that you see a lot in children going to daycare and schools. But it's obviously not the same strain that often has been seen to be a trigger of type 1 diabetes. Now, does everyone getting that infection get type 1? No. So there are a lot of unknowns there. But we have a very important program called NPOD, which is Network for Pancreatic Organ Donors. These are very generous people that have that have decided or given the permission to donate their pancreas once they're deceased, and that has been a huge source of knowledge for us to understand what goes on inside the pancreas of a person with type 1 diabetes and compare them with controls such as no diabetes or people with type 2 diabetes. Lots have been found from them. One of the findings there has been that we've seen viral infection in these pancreatic cells there. And so there is definitely a component of viral infection that triggers type 1 diabetes. The second source has been the gut microbiota. So it's not a matter of good bacteria or bad bacteria. It's a matter that people with type 1 diabetes and probably some other autoimmune diseases, not all of them, have an altered signature, altered composition of gut bacteria. And we know that the gut bacterium is very important in setting the immune tone, almost prenatally as well as postnatally, like immediately after birth. So that becomes an important component. The gut bacteria continues to play an important role in future metabolism, as in adulthood, for example. But there are some clues we have that can be a trigger for type 1 diabetes. There are other triggers that have been hypothesized with not yet a solid body of evidence. There have been a lot of things floating around vitamin D levels in exposure, a lot of things floating around a sudden shock in an individual, so on and so forth. Now, all of these things are being studied right now. Some interventions, a lot of research we have funded are now actually in translation. For example, we supported work with a viral vaccine in Finland that has now been pursued in a clinical trial to see if the vaccine can prevent type 1 diabetes. Wow. There are clinical trials ongoing with relying on information we have learned in gut microbiota. There are trials ongoing right now. So that's where these triggers are, and that's what we're trying to learn from, and then discover therapies that can, as I mentioned at the beginning, delay the onset of diabetes, ideally even prevent it, or even if someone that's newly diagnosed, can we have these interventions to change the course of the disease or disease modifying therapies, where you stop, slow down, or ideally even reverse, set the clock back, in people that have the disease already. And that's the mission of JDRF, essentially.
Yeah, it's interesting. You be thinking about.
Sorry to interrupt. Yeah,
No, good.
I did not answer your COVID question, because it slipped. I didn't mean to dodge it. It slipped my mind. So that's another enigma at the moment. I know the CDC reported at the beginning of January about an increased incidence of new onset diabetes. Other smaller groups have suggested that. At JDRF, we're looking at the data. We've been a science-driven organization. We are data-driven here. Right now, we don't have enough information to know whether the SARS-CoV-2 virus is directly or even indirectly causing an increase in type 1 diabetes, such as a viral, viral point of entry and a direct mechanistic, either through entering into some cells that will then cause type 1 diabetes or imbalancing the cytokine or the immune milieu there and doing it. We don't have enough evidence. What the CDC reported was from claims data, and that the claims data they reported were in people below 18 years of age, and that did not talk about not only about type 1 versus type 2, that didn't have much of a look back in the data about whether these people were predisposed, pre-diabetic, or anything else. So it's a little hard to glean into that data set, but JDRF is feverishly pursuing with other registries around the world, such as in the UK, Germany, US as well, and Australia, Israel, as elsewhere, where we are actually following up with these registries to look at all the data set and make a data-driven decision whether the SARS-CoV-2 virus is directly or even indirectly causing type 1 diabetes. There is no doubt that people that already have the disease, if they get an infection with SARS-CoV-2, the severity or the outcomes are poorer than in people without diabetes. There is no doubt about that. It's unequivocal. The severity of the disease, death and hospitalization is 3 to 4-fold higher in people with type 1 and type 2 diabetes once you get infected. But whether the infection is causing type 1, I think the jury is still out.
Got it. Yeah, it's always interesting how, as you were saying, the whole microbiota situation once again, and how these trillions of organisms that live within us, what they're doing to not just the adaptive, but the innate immune response, and how people with such and such infection may be preventative from getting something out. It's a fascinating domain, and it's great to hear that you're involved in thinking about it as well. That's sort of the main thing I wanted to profile there. That sort of the all-encompassing nature of how you look at this disease. It's a very refreshing. Let's put it that way, and very elegant approach that you take to looking at this. So it's very nice to hear it. So I want to just come full circle now. Obviously, I'm honored to be talking with an expert, a Ph.D. in biochemistry, microbiology, especially in translational medicine. But as you were saying at the very beginning of this, 50 years ago, a lot of this got started by everybody putting one foot after another, and the walks, which I've been part of here, as I said, in the Philadelphia area, and I know you've written about this as well. The importance that although there's all this cutting edge, bleeding edge science going on, we still got to get out there and do the walks and do the advocacy. Say a few words about the walks, if you would.
Oh, that is the core of DNA. That is the base. That's the base of the pyramid here. I mean, as I mentioned at the very beginning, we are representative of the people with type 1 diabetes and their families. We were not only founded by them, we're governed by them, we're supported by them through grassroots advocacy. That is everything that JDRF is about. And so, we need to support our research and our efforts in advocacy as well. And a lot of this almost entirely comes thanks to grassroots advocacy and fundraising. And some mechanisms we have are the walk. The walk is our pride, our point of pride, that we pride ourselves in. We also have, galas and auctions and other fundraising avenues, planned giving, and many other things that help us raise the resources we need, raise the dollars we need to support the critical research and advocacy efforts. And let me not, I didn't get a chance to bring it up. And the important point is that JDRF has internal bench strength that covers the entire pipeline of therapy development. It's not just the basic and the translational research. We have a dedicated set of experts that have regulatory expertise, that have health policy expertise. And then certainly, you know, once therapies are approved, we need to be supporting education and awareness around it. So there is access to the therapies. There is adoption of the therapies. Importantly, there's adherence to the therapies.
Yep.
Because no therapy is going to be a magic bullet.
Yeah.
So, you know, these therapies have to be affordable, have to be prescribed by the physicians, have to be adopted by the people with diabetes, and have to be adhered to by them. So it is part of a very important interconnected, interdependent network of things that happen here. But yes, the walks, you know, that are our chapters and our regions around the country, and there are parts around the world as well that support us to get there here. And it's not just a source of revenue, it is our point of pride. Yeah. It's the important family that gets us through. That's how we were founded. That's how we are, you know, that's how we lead every day. And that's, I think, that is what is going to get us to the finish line.
Absolutely. Yeah. I know it was near and dear to you, so I just wanted to bring it up as sort of a way to go full circle with all this. So
definitely.
Yeah, it's a fascinating set of initiatives, Sanjay. I could be continue to follow you, rooting you on all along the way, and getting us so close to these final goals of curing this dreaded disease. Really amazing stuff. For everybody that is going to be listening to this episode across the various podcast networks or watching on the YouTube channel, once again, you've been listening to the amazing Dr. Sanjoy Dutta, Chief Science Officer at JDRF International, doing really amazing things to cure and improve the lives of those currently living with type 1 diabetes. Sanjay, I want to thank you for taking the time out of your schedule to talk to us for a while. Obviously, thank you for everything you're doing there at JDRF. And as we say on this show, thanks for creating a better tomorrow for everyone out there living with this dreaded disease. Really fascinating story.
Thank you, Ira, for inviting me, and thank you to everyone that has been able to listen to this.